My name is Prisha Rai, and the title of my research is Utilizing a Novel Variational Autoencoder Pipeline for Tau Inhibitor Screening Validated in Drosophila Melanogaster Alzheimer's Models. Alzheimer's disease is a progressive neurodegenerative disorder impacting over 50 million worldwide. Currently, there are only 3 FDA-approved Alzheimer's drugs, all of which are acetylcholinesterase inhibitors which target the accumulation of amyloid beta plaques in the brain. There is a critical need for Alzheimer's treatments that discuss the other underlying mechanisms of Alzheimer's, such as the abnormal aggregation of the proteins known as tau, which current therapies do not target. In Alzheimer's, the tau protein becomes hyperphosphorylated, leading to the formation of neurofibrillary tangles, a hallmark of the disease closely associated with neuron degeneration and cognitive decline. According to recent studies, including findings published by the Journal of Alzheimer's Disease, tau pathology strongly correlates with the severity of dementia, even more so than amyloid beta levels, suggesting that tau is potentially a more direct mediator of neurodegeneration. The objective of my study is to employ a novel variational autoencoder pipeline to generate and screen molecules that could potentially serve as therapeutic agents against tau protein aggregation in Alzheimer's disease, using deep learning to accelerate the drug discovery process. We start off with the computational aspect of my project, which leverages a variational autoencoder and an LSTM-based property predictor. There's 2 parts to the variational autoencoder pipeline. First, the encoder takes in a chemical input in a tokenized format known as a SMILES, and from here, the SMILES is converted into a less dimensionalized continuous vector or latent space. And from here, from the latent space, the continuous vectors are converting back into a molecular representation in the SMILES format, and this output gives us a set of new predicted chemicals that have similar properties to the molecules in the training set. For the testing set, I went ahead through various publications on known tau inhibitors and compiled my own database of 70 known drugs that inhibit tau aggregation. The synthesized dataset has the same parameters as the initial dataset, namely QED, log-P, and SAS. The second neural network, the property predictor, predicts the solubility, synthetic accessibility, and quantitative estimated drug likeness score of the generated molecules by sampling this same latent space. This is a SMILES-based LSTM deep learning model, which uses multiple dropout layers to prevent overfitting, as well as dense layers to further process the model parameters. For the VAE framework, I tested out swapping the vanilla GRU layers in code segments with the bidirectional GRU layers and then bidirectional LSTM layers to see which outputted the least validation lost after 80 epochs. It was found that the bidirectional GRU layers showed the most idealistic asymptotic behavior, and therefore that was the layers we chose to go forward with in terms of the computational screening pipeline. To give a summary of the pipeline, we start off with, again, the variational autoencoder model, which was the first neural network built. This generates molecules, and then those molecules are ran through the second neural network, or the property predictor, and from here, molecules are filtered based on ranges deduced from the current FDA-approved Alzheimer's drugs, and from here we ended up with our final novel screen molecules, shown best here by these 3. The molecule we chose to test empirically upon the computational aspect was molecule 3, shown here, which had the highest QED value of 0.82, and this molecule is known commercially as methylene blue. Upon a literature search, it was found that a mechanism known as liquid-liquid phase separation is the mechanism by which methylene blue is able to inhibit tau, shown in the diagram here. The methylene blue clusters bind to the tau protein and cause a phase transition, causing the decrease in the neurofibrillary tangles. For the biochemical analysis in vivo, I quantified RNA by conducted a brain extraction of the Drosophila. The 3 groups were wild type with no methylene blue, Alzheimer's mutated with no methylene blue, and methylene blue mutated Alzheimer's flies. These flies were then anesthetized and decapitated, and their brains were very tediously extracted for a sample size of 24 brains. Standard protocols were followed for the subsequent RNA extraction and purification, and from here, spectrophotometry was used to find the RNA concentration values. In the graph shown here, we see that for the wild-type control values, the RNA concentration is around the 260 to 270 range, which is what's expected for flies of this nature. Also seen by the graph, we see a significant decrease in the RNA concentration with the Alzheimer's mutated flies, which is also proven by the statistical analysis shown here that this is in fact a statistically significant drop. What I do want to focus on though is this increase from the Alzheimer's mutated to the Alzheimer's mutated flies with methylene blue. Although by the graph we do see that there is a marked increase in the RNA concentration, this unfortunately was not a statistically significant value. I attribute this to my relatively low sample size due to the sheer tediousness of dissecting fly brains. In the future, I believe that this experiment could be improved by 1, increasing the sample size, and 2, using a more reliable method of RNA quantification, perhaps qT-PCR. Thank you very much.